we can start. Hello, everyone. Thank you for joining our webinar titled Qualitative and Quantitative Analysis, GMP Compliant, of Intact and Digested Proteins by LCMS-MS, a case study. Presented by Neutron, I am Caterina Cappuccini, Quality Control Manager of Neutron Pharma Lab, and I will be your host for today's session. If you have any questions in the middle of our presentation, feel free to use the question and answer section or the chat box. We will address those questions towards the end of our program. Before we dive into our main agenda, let's introduce our speaker for today. Dr. Sofia Magli graduated in chemistry and pharmaceutical technology from the University of Ferrara in 2019. In 2020, she joined the Neutron Pharma Laboratory in Modena, where she worked for 3 years on the development of LCMS methods for nitrosamine and nitrose API. She's currently working in the role of biopharma development project leader. This is the agenda for today, where I will start with a brief introduction of Cotecna, Neutron, and Neutron Pharma. Then Sofia will present her work, and after that we will discuss together some questions. Before to leave the word to Sofia, I will give you a brief introduction about our company. Cotecna is a leading provider of testing, inspection, and certification services. We offer solutions to make supply chain safer and more efficient for our clients. We are a Swiss-based company headquartered in Geneva with offices and labs across the globe. Today, we now have over 7,300 employees with 140 offices and labs, providing expertise across our 5 key sectors, namely food and pharma, agriculture, metals and minerals, system certification, and consumer products. This year, Cotecna is also celebrating its 50-year anniversary. Please, if you are interested, follow us on our social media for more information about our journey and key milestones for the past 50 years in the testing, inspection, and certification industry. Neutron is part of Cotecna. Neutron is a global player in the analytical sector for chemical, biochemical, microbiological, physically, and sensory of food products, feeding, supplements, food contact materials, cosmetics, and non-pharmaceutical products. Was founded in 1975. Today, the company boasts almost 50 years of experience and more than 5,500 employees providing high-level technical support to clients. One of Neutron's most important characteristics is to have all its departments located in a single operational site that span over 10,000 square meters of laboratories, as well as technical and commercial departments in support of our analytical activities. Neutron Pharma, that is part of Neutron, was born in 2015 and is a specialized GMP analytical services that, in adherence with the main international standards, provides chemical and physical tests and supplies high-level technical support to many of the world's major pharmaceutical companies in the field of GMP analytical service. We offer a complete chemical analysis service for the safety and quality of the entire pharmaceutical sector, API, drugs, excipient, raw materials, and packaging according to the GMP standards. Thanks to the concentration of know-how and technology, Neutron Pharma guarantees the highest level of quality and reliability of analytical data, as well as immediate assistance for our clients to meet any new pharma sector alert or regulation. Neutron Pharma is authorized by Italian Medicines Agency, FDA, Ministry of Health for the veterinary drugs and for drugs and psychotropic substances. At Neutron Pharma, we are able to offer a wide analysis portfolio on different matrix, including drugs, API, excipients, and packaging primary materials. Perform analysis according to pharmacopoeia as European, Japanese, USP, Chinese, and development and validation of analytical methods with different analytical technique like GCMS, LCMS, ICP-MS, and LC cutoff. Now I will leave the floor to our speaker, Sofia Magli.
Thank you, Caterina, for your introduction. So let's dig right in biopharmaceutical field. Are defined biological drugs, compounds that contain one or more active pharmaceutical ingredient produced or extracted by biotechnological methods from a biological system of human, animal, or microorganism origin. Belonging to the category of biological drugs are hormones, enzymes, blood products, serum, vaccines, immunoglobulins, and monoclonal antibodies. While classical drugs have small sizes, which is they are called small molecules, and a lower degree of complexity that make it possible to obtain them through standardized and reproducible chemical synthesis, the biological drugs are larger and have a higher level of complexity of the molecular structures, so they cannot be synthesized in the same way. They are drugs, mainly proteins, that contain one or more active pharmaceutical ingredients produced through or extracted from a biological system that is a living organism. I'd like to illustrate the importance of drugs based on biotechnology by showing evaluates pharma's forecasts. The importance of biological drugs will continue to grow in the coming years. Biological products will account, in fact, for 37% of total prescription and OTC sales in 2026. This is a very high figure compared to 30% in 2020. By 2026, biotechnology will account for more than half the 100 top-selling drugs. And according to Evaluate Pharma's projections, 51 out of every 100 drugs will be biotech-based. These products will generate 57% of the sales from this group, speaking to the higher price that biotech assets tend to command. The increasing success of this category of drugs is related to their ability to control or treat previously untreatable disease, in addition to their enhanced biocompatibility. The reference guidance document for biological products at present is each guidelines topic Q6b. Characterization of biological products includes the determination of physiochemical properties on the primary structure of the proteins, biological activity, immunochemical properties, purity and impurities, and in this case, impurities can be process or product related by appropriate techniques. The guidance underlines also the importance of using reference standards for testing batches of production. In fact, the product must be compared to an appropriate standard. For drug applications for new molecular entities, it is unlikely that national or international standards are available. Therefore, the manufacturer should have established and appropriately characterized in-house primary reference material prepared from representative of, of production batches and clinical materials. These in-house working reference materials used in the testing of production batches should be calibrated against the primary reference material. If the manufacturer cannot provide a comparison standard, the approach is to perform the analysis on almost 3 different batches if the analytical technique is a high precision technique, such as a QTOF LCM SMS detector. Otherwise, even 5 or 6 different batches, if other analytical techniques are used, must be analyzed to define a reliable product profile to be used itself as reference. The identity tests should be highly specific for drug substances and should be based on unique aspects of its molecular structures. Therefore, the main request for analysis for these kinds of products concerns the structural characterization of biological drugs. Currently, Neutron is focusing on proposed 2 kinds of activities for primary structure characterization. The first one is the confirmation of the molecular weight and the confirmation of identity of proteins or monoclonal antibodies or peptides performed on intact mass of the protein with top-down workflow in this case. While the second one is the amino acid sequence confirmation with peptide mapping technique and a bottom-up workflow approach in this case. Other proposal for structural characterization of proteins may involve confirmation of amino terminal and carboxy terminal sequences, the determination of glycosylation site, disulfide bridges analysis, and still again, analysis of host cell proteins in monoclonal antibodies. The reference guideline for validation method activities instead is the EACH guidelines Q2R2. The legal effective date of this guideline is 15th June of this year, a few days ago, but a draft version had already been available online for several months. And for the setup of our validation approaches in the biopharma field, we drew on this guideline. In Annex 2 of the guideline, it is interesting to note that precise parameters are established for several analytical techniques that were not mentioned in each Q101, such as ECPMS or ECPOES for the elemental impurities analysis, particle size distribution analysis, quantitative PCR, and also surely biological assay. Validation in this case, in our case, hasn't the classical approach of each Q101. The development of the validation methods have, in fact, the main purpose of identifying the structural characteristics of the protein or antibody analyzed. The only test actually required for validation is the specificity test, which mainly involves checking the absence of interference substances in the sample compared with a blank sample solution. The guideline also emphasized the importance of using orthogonal procedure comparison and different techniques for the consistency of the results generated, for the results obtained. From the research stages through development and process control, it is essential to have orthogonal analytical techniques for confirmation of the characteristics under study. Because this level of information becomes even more important in the development of biosimilar drugs. Where it is important to characterize the molecule of interest and assessing with accuracy and sensitivity any differences from the innovator product. Our general validation approach to dealing with an analytical request starts with setting the specification range in accordance with the customer. Then we write an analytical protocol with all acceptance parameters to be achieved. We share it with the customer in a draft version for their review, and once it is signed by and approved by both parties, neutron part and customer part, we can start with the method setup and optimization of the sample preparation. This step should be repeated on each different kind of metrics. When the method works, we proceed to perform all validation tests as set in the protocol. The results obtained are analyzed and compared with the acceptance parameters of the protocol. If all is okay, all data obtained are collected in a final validation report that is shared with the customer. Is important, in my opinion, to emphasize here the importance of this final analytical report for sharing results with the customer. That are because these results are mostly qualitative, so a certificate may be reductive in this case. Turning our attention now to methodology validated for intact protein identification and molecular weight confirmation. In GMP compliant, it was firstly necessary to write an internal protocol to perform validation activities on a sample used as an example for the development of the general methodology to be used for intact protein identification and for the confirmation of the molecular weight of proteins and peptides. Our methodology development was based on 4 different tests with related acceptance parameters, which you can see in the graph on the left of this slide. The first one is verifying the absence of interferences by injecting a blank sample solution acquired by the same method of protein sample analyzed. The identification of the protein product wasn't impacted by the presence of other substances, impurities, degradation products, related substances, or other components likely to be present. The second one was a specificity and selectivity test. The profile obtained for the product was highly specific for the protein used for method development validation and based on unique aspects of its molecular structure. In this case, if available, reference standard or in-house reference standard sent by the customer is used for comparison with the sample. It should be checked the alignment of the retention time referred to the product peak in the standard solution and compared it to the retention time referred to the protein peak in the sample. If the standard is not available, the test consists of evaluating the molecular weight obtained in the sample by comparing it with the expected and theoretical molecular weight. For accuracy test, the mass error obtained was evaluated. The mass error was obtained from this formula by subtracting the theoretical mass weight from the measured mass and dividing by the theoretical mass. The result obtained is then multiplied by 10 to the 6 in order to obtain the results in terms of ppm. We set in our validation procedure at maximum 5 ppm for large proteins and 2 ppm for peptides, the setability. The difference comes from the fact that peptides have lower molecular weights, and we can, for this reason, accept lower mass error. At the end, the instrumental precision test is, in this case, a repeatability instrumental evaluation performed on 6 or better, even better, 10 different injections of the same protein, extract or, if available, of 6 or 10 different injections of the same standard preparation. The relative standard deviation percentage was then calculated on different mass error found, and this value obtained was not more than 20%, the acceptance parameter that we set for instrumental precision test. Let's move on the methodology validated in Neutron for amino acidic sequence confirmation by peptide mapping technique. Regarding this methodology, the validation method procedure is ongoing. This validation procedure set by our internal protocol is based on 3 different tests with related acceptance parameters. The first one is, again, the absence of interferences that should be verified in a blank sample solution digested with the same digestion protocol and in the same digestion conditions as the sample solution. For specificity and selectivity test evaluation, it is important to introduce the concept of coverage. That is how many peptides could be covered of the protein to be identified. Starting from the amino acidic sequence of the intact protein to be identified, that is always given by the customer, the protein is digested and all the small peptides obtained are acquired after digestion of the protein. The coverage indicates how much of the starting amino acid sequence can be confirmed to exclude, in order to exclude, for example, post-translational modifications. Guidelines have no clear indications to coverage value that can be considered okay for an analysis of peptide mapping. Internally, we set acceptable coverage value if it is not less than 80%. The repeatability the last test should be evaluated on 3 independent digestions of the same protein. Coverage obtained for every different sample preparations should be more than 80%. And relative standard deviation percentage should be calculated on the 3 different coverage values and should be not more than 20%. Our familiarization tests started from a standard certificated of a protein of intact bovine serum albumin. The most critical step for peptide mapping analysis is the digestion procedure of the sample. Therefore, we tested and screened different brands and types of protein digestion kits. Finally, we choose a Pierce digestion kit because it already has within it the compounds needed to perform all the digestion steps. Because of the speedy of digestion it guarantees and the high performance of digestion obtained with that kit. The kit already has all the reagents used during all steps of intact protein digestion inside. In the first step of enzymatic digestion is used DDT in protein excess in a ratio of about 1 to 10 in order to reduce intra and inter disulfide bridges and denaturate the sample at high temperatures. Iodoacetamide is then added to alkylate the sample in their conditions. Digestion occurs with the trypsin enzyme, in this case, in our case. Trypsin cleaves exclusively C-terminal to lysine and arginine. We choose trypsin among many digestion enzymes due to its characteristics of cleavage. It creates neither too small nor too large peptide fragments. Arginine and lysine are amino acids consistently distributed in peptides, which allows for fairly regular peptide fragments. After tryptic digestion, small peptide fragments from the bovine serum albumin intact protein are generated. The digested sample is then analyzed with the LC-MS QTOF instrument after the development of the correct instrumental methods for mass part and development of correct chromatographic conditions. A specific software is used that allows the theoretical amino acid sequence provided by the customer to be entered and compares the matches with all peptide fragments obtained during acquisition of the digested product. The acquired data are reprocessed in order to obtain a percentage of coverage of the protein. To monitor the protein digestion process, we also introduced a positive check. We purchased a standard of peptide fragments of already digested bovine serum albumin as a check of the digestion processes, the poor or excessive one, and optimization of the analytical method. For identification tests, in this case, demonstrate the capability to identify the compound of interest based on unique aspects of its molecular structure is a critical aspect. The comparison between the standard digested in neutron versus the standard already digested with the peptide fragments is a checking of positive results, as also suggested by the guideline. To be even more confident of the digestion of the bovine serum albumin sample in this case, we have also introduced a negative result control by comparison of the digested sample with an intact undigested albumin sample. An interesting case study was also one of the first approaches for us to validate a method for intact protein identification, or better, in this case, for identification of an intact small peptide and insulin. The request was from a customer who is developing a new product this peptide, and the client needed to have a validated analytical method specific for this product that would allow them to standardize the identification of the peptide throughout all process of synthesis for the analysis of different batches under different stability conditions. We started by collecting the preliminary essential information provided by the client. The first one was the theoretical molecular weight of the peptide, in our case equal to 3,751.20 Daltons in accurate mass. The second one was the specification range of the measured molecular weight versus the theoretical molecular weight given by the customer, equal to 1 Dalton more and 1 Dalton less. Then the request to have availability of an in-house reference material to be used as reference, as standard. In our case, the customer had availability of the standard. And at the end, the research of the best elution solvent for the product, in our case, methanol. Our first approach to the method development was the classical approach we always have for small molecules analysis. We started with the method setup as a direct injection in LCMS instrumentation. In the table, you can see the top 10 masses obtained from direct injection acquisition in LCMS instrument are ordered for intensity of their areas. And in the left column, you can find their masses expressed as the ratio between mass and charge. Of these 10 masses, only 2 masses can perhaps be traced to a tricalicate of the peptide, and they are highlighted in blue color. The problems associated with the method were immediately clear. The first problem was an instrument with an inadequate performance for the identification of proteins of that size, with too limited mass range from 200 to 1250 Daltons, expressed as a ratio between mass and charge. The second problem was that we had underestimated the structural complexity of the peptide under analysis. Consequently, the results obtained were not within the specification range set and required by the customer. So we transferred the method setup to Zenotov instrumentation, avoiding direct injection for the validation of the method. We started with the development of the chromatographic method. We operated with a superficially porous particles core shell column, peptide-specific column with silica C18 derivatives packing in order to obtain an intermediate selectivity for small and hydrophilic peptides. This allows us to achieve a higher retention and selectivity and improve the peak shape. We also added formic acid to the mobile phase A and mobile phase B, the organic and inorganic one, and peak shape was dramatically improved. We also optimized the phase elution gradient to obtain a slower gradient where the percentage of organic phase reaches 30%. That is the percentage that corresponds to the exit of the peptide through the column. We set the column temperature at 50 degrees to improve the peak shape, tightening it and decreasing the classical tailing phenomenon of the acquisition of high molecular weight proteins. And at the end, regarding obviously the mass part, the Xenotroph LCMS MS instrumentation has an higher mass resolution that LCMS instrument, and a wider mass range of acquisition between 2000 and 4000 between 2000 to 4000 Daltons expressed as a ratio between masses and charge. We performed validation with the optimized methods, and the results obtained were very good. The blank sample acquired, obviously with the same method as for the sample, had no interfering peaks at the retention time of the peptide under analysis, and absence of interferences was verified. The retention time of the peptide in the sample and in the standard solution provided by the customer was the same, equal to 14 minutes and 60 seconds in the standard acquisition and 14 minutes and 61 seconds in the sample acquisition, as you can see from the chromatograms in the slide, in the current slide. The alignment of the retention time referred to the product peak in the standard solution and in the sample makes it possible to say that the specificity test was complies. The molecular weight of the convolute mass found in the sample was equal to 3,750.90 Daltons in accurate mass, and the molecular weight found was within the required specification range and matched the molecular weight of the peptide on the standard solution, equal to 3,750.90 Daltons too. In conclusion, let me sum up my main points. Biopharma is growing steadily and will be an important topic in pharmaceutical processes in the future. Surely, new guidelines are emerging to provide guidance on the validation and testing process for the biopharma world as well. The characterization of complex proteins is very different from the classical approach used for small molecules, and validated methods are useful for standardizing the manufacturing processes of biologics and making easier analysis of all batches throughout the protein synthesis process. And at the end, the technological effort required for the analysis of biopharmaceutical products is much greater than the effort required for the validation of methods for small molecules, partially because there are often no reference standard certificated. In conclusion, I want to say that characterization of therapeutic biological drugs require more technological and analytical efforts than are required for the development of characterization of chemical molecules. These differences are reflected in the increasing attention and severity that regulatory agencies are placing on the regulation of biological products that in the past were perhaps insufficiently characterized. Thank you for your attention.
Thank you, Sofia, for your very interesting presentation. We will open the floor now for a question and answer section. We have received during the registration process a few questions, And we start with the first one. Is it possible to perform a quantitative validation on intact proteins?
Yes, it is possible. A quantitative full validation is a request that, at least in my experience, occurs less frequently than a qualitative determination in order to confirm the molecular weight of the proteins. However, with our software and on Genotop LCM SMS instrumentation, it is certainly possible to perform it. More than ever in this case, the best case is to have a standard or an in-house reference standard available to construct a calibration curve. And even better would be easy to perform validation in a quantitative way with the possibility of using a placebo of the sample. In this case, the validation approach can be used with classical linearity, accuracy, intermediate precision tests as for small molecules validations. When placebo is not available, all tests can be performed by directly spiking the starting matrix protein with the standard or a reference standard provided by the customer in order to obtain a calibration curve. The curve is then used to recalculate the content of the sample unspiked. When the reference standard is not available, the calibration curve is constructed from progressive dilutions of the matrix itself, with no title as a matrix calibration curve. For example, by acquiring the sample as it is, 100% diluted to 80%, 50%, and 10% progressively. The intact quant software of the QTOF LCM SMS system is used for quantitative analysis. It is important to underline that in this case we are discussing about title as far as API sample. Usually based on, as far as API sample is concerned, not protein content in finished drug products. The quantity of the drug substances usually based on protein content mass should be determined using an appropriate assay.
Thank you. And we have another question. What are the difference between the two analyses for protein characterization, intact protein identification, and peptide mapping?
Okay. The characterization of a biologic drug is given by the use of several orthogonal techniques. These include confirmation of the identity of a protein by analysis of the mass, monoisotopic or average mass of the protein, and confirmation of amino acid sequence by peptide mapping technique as described in the guidelines. Each technique has distinct methodologies and applications. The intact protein identification involves analysis of the entire protein molecule proteins without prior digestion into smaller fragments. For this reason, it requires minimal sample preparation compared efforts compared to peptide mapping analysis, as the protein is analyzed in its intact form. Information provided by this kind of analysis are the accurate molecular weight of the intact protein. It can help identify different isoforms or variants of the protein. And the main applications of intact protein identification are the identification of protein isoforms, and it's ideal for rapid screening and characterization of proteins in their native form. For peptide mapping analysis, the protein is digested into smaller peptides and involves a complex sample preparation with multiple steps. This technique provides detailed information on the amino acid sequence, including post-translational modifications, allowing also the identification of specific peptides and their locations within the protein. We can provide information on the location and type of post-translational modifications, such as phosphorylation, glycosylation, and acetylation. Main applications of peptide mapping analysis are in-depth characterization of protein structure, including primary sequence and post-translational modifications, and in biopharmaceutical development is critical in the development and validation of biopharmaceuticals ensuring.
Thank you for your answer. And we have another question that is it essential to have a reference standard for the execution of biopharmaceutical analysis for batch release?
No, it is not exhaustive. The manufacturer is required to provide an in-house reference material. If the manufacturer is unable to provide it, can be analyzed 3 different batches of the product in the analytical technique. If the analytical technique is an high precision technique, such as a QTOF LCM-SMS detector. Otherwise, even 5 or 6 different batches are analyzed if other analytical techniques are used. Different batches must be analyzed in order to define a profile to be used itself as a reference.
Thank you. I think we have some couple of minutes more for one last question that we have collected. What are the advantages of having a method validated on a specific product in order to standardize the processes?
In the biopharmaceutical field, having a method validated on a specific product to standardize the processes offers several advantages. Firstly, ensures that the process produce the desired outcomes with minimal variability and with an high quality and uniformity in every batch of the product. Validation provides also scientific basis for the process parameters and their control, helping in understanding the critical aspects of the process and allows for a better optimization. The standardized processes also, in my opinion, reducing the risk of failures and deviations and help to standardize processes and minimize the risk of variability. Surely reduce the incidence of batch failures, waste, and the need for rework, leading to cost savings. And at the end, validations required through documentation, which provides a detailed record of the process parameters, controls, and outcomes. In summary, method validation on a specific product for process standardization in the biopharmaceutical field is crucial for ensuring product quality, regulatory compliance, risk mitigation, cost efficiency, and overall process robustness, leading to safer and more effective biological products.
Thank you, Sofia, for your answers. As we near the conclusion of our program, we will send out an email in the coming days containing a survey form and the recording of this webinar. Additionally, for more news and updates about Cotecna, we will be providing a link to our newsletter subscriptions. You may also reach out to us using the contact information provided on your screens. We hope you find this webinar beneficial. A big thank you again to our speaker, Sofia Magli, for sharing her expertise, and to all of you for joining us. Have a great day, everyone, and see you in our next webinar.